Departmental Warren Lecture Series. Our speaker today is Alfred Spormann from Stanford University. Alfred got his PhD in 1989 from Marburg University in Germany. He then went on to, as a postdoc, to University of Minnesota. He worked for 1 year with Harry Hogenkamp in the Department of Chemistry. And then he joined Stanford University, one other postdoc, to work with Dale Kaiser. And he must have left a very good impression there because they hired him right away as a faculty member at Stanford in the Department of Civil Environmental Engineering. And since then, he's faculty there. He has also an appointment in chemical engineering. In 2013, Alfred has been elected fellow of the American Academy of Microbiology, and he was for several years editor of the Applied and Environmental Journal of Applied and Environmental Microbiology. And Alfred has several years, he served as director of the Woods Hole Microbial Diversity class, and then he is also the founder of the Hopkins Microbial Diversity class at the Hopkins Marine Station in Monterey, which also operated by Stanford University. Alfred's research interests are general processes in anaerobic microbiology. Yesterday, he gave a great presentation at the Biotechnology Institute related to bioenergy aspects. Today, he's going to talk about bioremediation aspects, but he also works on the gut microbial communities in the large intestine in this respect. So the presentation today is going to be on the organisms that breathe chlorinated solvents. So we use oxygen as terminal electron oxide. These organisms use chlorinated solvents. And the title is Microbial Life by Reductive Dehalogenation. So thank you very much, Alfred, for coming, and we are looking forward to your presentation.
Well, thanks, Sebastian, for the nice introduction, and I thank the department for inviting me to give a talk. I'm delighted to be here. First of all, I think it's a great department, and secondly, it's just like a coming home because, as Sebastian said, in '89, I was a postdoc just right across the street, and I think it was Millard Hall at the time, and I don't think Millard Hall exists anymore. That's how it goes, and the biochemistry department was there, and I had a wonderful time here as a year on being a postdoc in Harry Hogenkamp's lab. So I want to give you a flavor of the kind of questions we ask and the type of research we're doing with respect to reductive dehalogenation. And reductive dehalogenation is a theme that has been occupying us for at least 15 years. And the progress typically was accelerated by technical breakthroughs so that we could ask certain questions that we couldn't ask. And this topic, I think, is a lecture on persistence. Not only that this is a persistent compound in the environment, but also persistence in terms of pursuing questions so that ultimately you find some answers. And I understand that this is a broad audience, so I try to be general, and then for the more technically versed, I'll give you some specifics on some research that we are actually quite excited about. So I'm taking the view and perspective of a microbiologist, and I want to look at these reductive dehalogenations and specifically talk about the organisms that are involved in chloroethene degradation and give you sort of a flavor of what we think is the business plan of this organism. In a very simple way, you can think that every organism has a business plan. In as much as every business has a niche that it caters to, organisms have the equivalent. And I don't want to drive this anthropomorphic thinking too far, but at least it gives an idea of how to think about where the niches is and what sort of the business plan is of an organism. So here's the problem. Superfund sites in the US, there are almost 1,000 Superfund sites distributed around the US, and most of them contain chloroethanes or chloroethenes as contaminant. It's a big problem The legacy in terms of remediation is in the range of billions of dollars per year. And it's an important question because these chloroethanes threaten the quality of drinking water. So if you look at the most prevalent groundwater contaminants, they are chloroethanes and chloroethanes. And in many sites, specifically DOE sites, sites from which, and these chlorinated solvents were used for degreasing materials. It was used for degreasing jet engines by the military. It was used in DOE labs. And it was used as in the dry cleaning industry to dry clean clothes. And it was typically improper disposal and leaking storage tanks that released these dense non-aqueous phase liquids into the environment, into the groundwater, and then through a steady dissolution into groundwater ultimately caused prevalent contamination of groundwaters. So of these compounds, we have the chloroethanes, tetrachloroethene, which is abbreviated as PCE, or trichloroethene, and the tetrachloroethanes and tri- and dichloroethanes are the prominent groundwater contaminants. I'll talk about these individually, but the bottom line is the degradation of these compounds you cannot view independently because the degradation pathways intersect, and it has huge implications for how we are treating these contaminants. Well, the microbial strategy, or the effect of microbial degradation of these chloroethanes is by stepwise reduction of these carbons, which are oxidized here in a carbon-chlorine bond, to ultimately the harmless ethene. And ethene is the plant ripening hormones. If you want to have bananas or apples ripened, then this is actually induced by a hormone which is essentially ethene. And in this step, there's a stepwise reductive dehalogenation to lesser chlorinated compounds. And of particular importance is vinyl chloride, which is a natural intermediate, but it's typically also a bottleneck. It's a kinetic bottleneck that vinyl chloride actually seems to accumulate in many sites. What is more important is vinyl chloride is a known human carcinogen, whereas PCE and TCE are suspected human carcinogen. So in other words, if you have an incomplete microbial reductive dehalogenation occurring at sites, you convert a suspected human carcinogen into a known human carcinogen. So we focused most of our attention on vinyl chloride and the degradation pathway involved in vinyl chloride degradation. So I'm building now this slowly to a subway map, not of Minneapolis, but a subway map of degradation of these chloroethanes. Here I drew in sort of the type of organisms that are known to mediate this dechlorination of the higher chlorinated chloroethanes to cis-DCE or even vinyl chloride. And as this shows you here, this is actually a diverse group of microorganisms can catalyze these reactions. Whereas the most important step for the final detoxification and vinyl chloride degradation is done by a group of organisms called Dehalococoides. These are chloroflexi. It's one very specialized group of organisms that has so far this unique capacity. And in many sites that we see that have complete reductive dehalogenation, they contain this organism. And they also contain specific genes which we can monitor, specifically the vinyl chloride reductase, which we studied in my lab very extensively. There are other putative vinyl chloride reductases that are also found, but to a lesser extent. We also know about the genes and the enzymes involved in this first dehalogenation and the second dehalogenation step. They're called PCEA because it's a PCE dehalogenase and a TCA dehalogenase. So we have an idea about the organisms involved as well as sort of the key genes that are catalyzing this process. What makes things more complicated if you have mixed contaminants. So if there are contaminants coming from the chloroethane side, well, we know that tetrachloroethane is converted to trichloroethane at a relatively high efficiency, but there is also some conversion to chloroethanes. But more importantly, trichloroethene and dichloroethene can be converted to vinyl chloride by a dihalo type of elimination. It's an interesting chemistry, but it's also mediated by these reductive dehalogenases. And also in this part here, we have the more general group of organisms which catalyzes the dehalogenation of tetrachloroethane. But the final steps, again, is mediated by this very niche specialized group of dehalococoides. So the point being here, from a remediation point of view, you want to make sure, for example, the implication is if you have a PCE and TCE contaminated site, which also contains tetrachloroethane. If you start to focus on the remediation of this compound first, you may build up a toxic intermediate, which might be difficult to eliminate if the organisms and these genes are not present. And vinyl chloride can inhibit other processes. So when you design remediation strategies, you need to know what contaminants around, you know the metabolic interference of the degradation pathways, and what genes and organisms are present. So it might be favorable first to degrade the chloroethanes by stimulating this group of organisms, because if there's vinyl chloride accumulation, it does not prevent the degradation of this process. That's the kind of reasoning that you can deduce from these kind of studies. What is also important is to keep in mind that these organisms here have also a generally sort of distinct physiology, while these dehalococoides are more specialized to really this vinyl chloride and cis DCE. This group can use the higher chlorinated compounds. But these organisms, and this is now the group 1, the upper group that I showed you first, it's phylogenetically more diverse, but the organisms can also a whole suite of other compounds as electron acceptors for growth. It's not only the electron acceptor that is different, but also the electron donor. For example, these organisms can use acetate, hydrogen, butyrate, lactate, and so on and so forth. Whereas the dehalococoides type of organisms are specialized to use only hydrogen as electron donor and only chlorinated compounds as electron acceptors, as catabolic electron acceptors. So these organisms are much more niche specialized. So this reductive dehalogenation, which is at the key of the remediation, is, chemically speaking, an elimination reaction which reduces this carbon here to a carbon-hydrogen bond. The energy of a reductive elimination of chloride is around a minus 150, give or take, kilojoules per mole. So that's a decent amount of energy that can be used by organism. It is important to keep in mind that there are two types of reductive dehalogenases, so the enzymes that mediate this reaction. They are energy-conserving reductive dehalogenases, which basically means that mechanistically, organisms can conserve the energy, these minus 150 kilojoules per mole, of the reductive dehalogenation, for example, here of vinyl chloride, and use this energy for growth. So mechanistically, the enzyme works for energy conservation. And this works in the class 2 organisms, basically the dehalococoides. In the class 1 organism, you may have non-energy conserving reductive dehalogenases where organisms catalyze the same reaction, but where the strategy of the organism is not to use that energy that is released, but to use the now dechlorinated carbon compound as a substrate for metabolism. So it's essentially a way to access new nutrients which are tagged previously by being chlorinated. These two types of dehalogenases have some distinct differences. If we are zooming into the cell, what is really at the core of this reductive dehalogenation? It's one of the most, for the energy-conserving organism, it's one of the most simple modes of making a living because it just requires two enzymes. One is the reductive dehalogenase, which here is a vinyl chloride reductase, that eliminates this chlorine, and a hydrogenase enzyme that uses hydrogen as electron donor. We don't know exactly how they are coupled, but essentially these are these two enzymes that are sufficient. Somewhere in this process, protons need to be translocated across the cytoplasmic membrane and then are used in the membrane-bound ATPase to conserve this energy in form of ATP. We studied these enzymes in more biochemical detail, and I will talk a little bit about this. This enzyme consists of an unusual combination of cofactors, and it's unusual because the cofactor is a coenzyme B12. And for those of you who are familiar with coenzyme B12 biochemistry, something that I learned with Harry Hogenkamp here at Minneapolis, is that B12 enzymes typically catalyze group transfer reactions or generate radicals. But in this case, coenzyme B12 is involved in a net redox reaction. That's a totally new function. Moreover, this coenzyme B12 is found in the context of two 4-iron 4-sulfur clusters, which are involved in the electron relay to this active site. So it's really an unusual enzyme, this reductive dehalogenase. And you can find in the genome in the gene sequence these specific patterns of iron-sulfur cluster regions that are important for iron-sulfur cluster formation. And then there's typically another subunit, a very small subunit, hydrophobic subunit, which is the membrane anchor. So if you look at genomes, metagenomes, you can recognize fairly well these reductive dehalogenases. If you look at all reductive dehalogenases and compare the amino acid sequence of these reductive dehalogenases and do alignment, and you essentially see sort of that these reductive dehalogenases fall more or less into two major classes. There's this major class here that groups certain reductive dehalogenases together, and all these green dots are those reductive dehalogenases that are involved in energy conservation. So they belong to the dehalococoides group that dehalogenate vinyl chloride. On the other hand, these reductive dehalogenases belong to this facultative organohalide respiring organism. That's the organism that dehalogenate the first steps in this pathway. So on a molecular level, you can actually distinguish between these dehalogenases. And when we started to look at the genome of these organisms to learn more about the physiology of this organism and what implications the biology of this organism has for bioremediation, we noticed, and others, that the genome size of this organism is extremely small. It's around 1.4 megabase pairs, which is really at the lower edge of any free living organism. The genomes of other organisms are typically symbionts, endosymbionts, but not free living. So in other words, this is a really minimalistic microorganism. It became even more interesting when we looked in a comparative way at the genome composition. And this shows a comparison of 4 of the dehalococoides genomes that we have. In the meantime, we have 11, but the fundamental features is basically the same. So what you can see here, it's a small 1.4 megabase circular genome, where most of the genome here, everything in blue, looks pretty much parallel. The pattern looks the same, which basically indicates there's a synteny in the genes throughout the genome. In other words, 70% of the genome of different dehalococoides is identical. You would not find this actually in E. coli, where you have much more of a gene rearrangement and scrambling of genes. What stands out essentially are these 2 regions here, where you can see here the similarity in these 2 regions sort of collapses. And what are penciled in here in red are reductive dehalogenase genes, so the key genes of the organism. I should say, if you look in the genome, and also we know it from physiological experiments, the organisms are obligate organohalide respirer. There's no other business plan other than reductive dehalogenation. So they can't ferment, they cannot respire. It's a very specific, limited way to grow just by this reductive dehalogenation. Moreover, each organism here has up to 30 or 38 different reductive dehalogenases, meaning while the organism is very specialized to reductive dehalogenases, the different dehalogenases that are present in the organism allows the organism to access different halogenated substrates as electron acceptor. That's the theory. We don't know what most of these reductive dehalogenases are doing, but that's what we would basically predict, and we know also from gene expression studies. Now, the interesting part is that all this diversity that gives the organism the flexibility to adapt in the environment to different compounds is really restricted to two regions here. And if you look closely, actually, let me skip that. No, I do need this. If you look into this region, you see that these reductive dehalogenases are nicely aligned with respect to a specific gene. This is called SSRA, and that serves as an integration site for acquisition of genes. And what we find is that the modules that contain these reductive dehalogenases, so on a more general question is, how does the organism actually require and manage these different reductive dehalogenases? And the answer is, these are basically acquired by lateral gene transfer. And I'm just telling you how this essentially works. We find that there are genomic islands with reductive dehalogenase genes. So here we have the reductive dehalogenase genes for vinyl chloride degradation. And then there is an integration module which is just required to get this piece of DNA inserted into the dehalococoides. It's just like companies expand their range to produce new things by just buying up small companies. And the buying up of small companies more or less works always by the same mode. I don't want to put the anthropomorphizing further. So we know that the molecular fingerprints of that, and we know that these modules to bring in new reductive dehalogenase genes into the organism repeat over and over. So we have clear genomic signatures that show that. We also know that there's evolution going on within these two modules. In other words, there are different types and different paths how these reductive dehalogenases, specifically the vinyl chloride reductase, uses different vehicles to get into these dehalococoides. And that basically shows you that there's also a scrambling and an independent evolutionary history between the vinyl chloride reductase and different modules to get into different organisms. These are pure strains, and these are enrichment cultures from which we have a sequence. So these genes and these genomic islands evolved independently, which is interesting to think from a bioremediation point of view. So there is not a master reductive dehalogenase that does all the dehalogenation in the world. It seems to be site-specific that this module for, in this case, vinyl chloride dehalogenation evolved independently at specific sites. So that's important to keep in mind when thinking about remediation strategies. And this is just a general way. These are these islands, these genomic islands for vinyl chloride reductive dehalogenation with reductive dehalogenases here. And this basically explains why they are all in one locus and how the organism ultimately expands its substrate range by acquiring just different reductive dehalogenases. That's basically how the organism evolved. And that's essentially the interesting aspect is also we know, based on the molecular analysis that I don't want to get into detail, it's essentially looking at codon usage bias for all these different organisms. These are all different organisms here. And we ask that from cultures that can degrade vinyl chloride. We know that the acquisition of new genes always occurs here, and thereby pushes the other genes in this direction. So in other words, these are the most recently acquired genes. So now look where they are for vinyl chloride reduction. Here, and this is the strain that we work, it's the most recently acquired gene. This one does not have a vinyl chloride reductase. Here you see the vinyl chloride reductase, here you see the vinyl chloride reductase, and here you see the vinyl chloride reductase. So they are all the most recently acquired genes. In other words, these organisms that we find at sites that degrade vinyl chloride in the environment most recently acquired this biochemical capacity. Okay, so what do we know about these vinyl chloride reductases? We, as I said, these are an interesting class of enzymes. The organism grows slowly, has a doubling time of 1 to 2 days. For those microbiologists in the audience, the maximum OD is about 0.1. Strictly anaerobic, grows on hydrogen and vinyl chloride. How do you do biochemistry with that? Well, it was a dedicated postdoc, Jochen Müller, who in an heroic effort grew 200 liters of this organism collectively, anaerobically centrifuged the organism, and then did an anaerobic protein purification for a vinyl chloride reductase. And he managed to purify this enzyme. It was a heroic effort. We did few kinetic measurements, Km measurements. We identified substrate specificity, but that pretty much exhausted the amount of protein we had. And it took about 10 years of efforts to try to come to a protocol that allows us to heterologously express these reductive dehalogenases. Many enzymes can be heterologously expressed. We tried it for many years, and it was another postdoc, Anudaman Padasarati, who actually isolated and was able to heterologously express the vinyl chloride reductase. And the trick was, don't try to express active enzyme in the cell, but use in vitro reconstitution to ultimately assemble the active holoenzyme. So we started with a his-tagged, MBP-tagged vinyl chloride gene. We overexpressed it in E. coli. We had inclusion bodies. We solubilized the inclusion bodies in 8 molar urea. We purified the resoluble, the soluble protein on a nickel column. And then we dialyzed out the urea, which led to refolding of the protein. And then we added ferrous iron sulfide and B12, and that reconstituted the active enzyme. And that was one of the first breakthrough, the first breakthrough that we had was the genome sequencing, because actually we could see what's going on in the organism, what it in the past. This was the second technical breakthrough, because now we had all of a sudden milligram quantities of the enzyme. So the heterologously expressed protein just looked like the wild-type protein that we isolated 10 years earlier. The kinetic parameters of the heterologously expressed protein had the same Km and Vmax. But we discovered an important, since we had enough protein to study, we discovered that the heterologously expressed vinyl chloride reductase catalyzed also the conversion of 1,2-dichloroethane to ethene. So that's a dihaloelimination, which basically means that intersects the pathway of chloroethane degradation to the chloroethene degradation. So in other words, the enzymes, some of these enzymes actually can mediate this step. So they might be linked in the same organism. We then use this enzyme, the purified enzyme, and collaborated with David Britt at UC Davis on APR spectroscopy to see if we can see some features of the enzyme. In the oxidized state, we have essentially here a cobalt-2. So the next few slides will be a little bit technical, but I'm just excited that we can actually do these kind of experiments. And we saw that the enzyme, as we identified, has a cobalt-2, and it has a hyperfine splitting that I'll talk about in a minute. When we reduce this with an electron donor, in this case titanium citrate, we reduce these two iron-sulfur clusters. So we see this actually in the EPR spectrum. And in vitro, you add titanium citrate, and then you get in vitro activity. And then if we add methyl biologen, which is a single electron shuttle, we can eliminate this cobalt-2, which is then reduced to cobalt-1, because we expect that cobalt-1 is actually what is catalytically active. And you need methyl biologen for activity. And if you simulate an iron-sulfur cluster spectrum, you basically get that. So the enzyme, the heterologously expressed enzyme, just behaves like an enzyme that is active in taking up electrons, reducing the iron-sulfur cluster, and also reducing cobalt-2 to cobalt-1, which is important for catalysis. This hyperfine splitting in the enzyme here, it's basically this region here, raises an interesting question where this hyperfine splitting comes from. And we use basically adenosylcobalamin. This is this molecule that we are essentially looking at here. And we showed that if you eliminate this lower ligand here, this dimethylbenzimidazole ring, if you eliminate this, you lose this hyperfine splitting. If you use cyanocarbonamide or aquacarbonamide, we actually lose this EPR structure, which basically tells us in the active enzyme, we have an interaction of a nitrogen which comes from dimethylbenzimidazole and not from a protein histidine that interacts with the cobalt here to give us this signal. This has some catalytic implications. In the meantime, there came a crystal structure out from the non-energy conserving reductive dehalogenases. And this is essentially the protein. It's a very simple protein. It's about 550 amino acids. You see in red the B12 cofactor, and in yellow here the iron-sulfur cluster. And if you just strip away the protein and just look at the active site, and I think even in the back you can see it, you can see here this is the dimethylbenzimidazole, this is a chloride, a chlorine ring, here a planar. The iron-sulfur clusters are in proximity where electron transfer is possible. And it looks like the the distal iron-sulfur cluster cannot directly interact with the chlorine ring relative to the proximal iron-sulfur cluster. Again, this is in the non-energy conserving reductive dehalogenase. So having that information and our EPR spectroscopic data, here's the model how we think that these reductive dehalogenases work. We essentially have vinyl chloride, and then through a cobalt-1 that we can see, well, it's EPR silent, but we know we have, that presumably makes a nucleophilic attack on this carbon, eliminates chloride. This leads to homolytic cleavage, presumably to make a vinyl radical, and then cleaves this to have a cobalt-2 back. The cobalt-2 is reduced by one iron-sulfur cluster with one electron. The reduction of this vinyl radical either may occur from an electron of a second iron-sulfur cluster, or it may occur through the same iron-sulfur cluster And this is essentially postulated based on the proximity of these two iron sulfur clusters relative to the Korean ring. So that's a working hypothesis. That's not proven. But since we now have protein, sufficient protein, we can do this structure function analysis. I want to turn the page now and talk about, again, these reductive dehalogenases. We know, sort of on a global level, we have two classes of reductive dehalogenases. These are the ones that we have been studying in Dehalococoides, and these are the ones actually from which we have the crystal structure, and these are found in the class 1 type of organism. What you would like to do as an engineer is make use of this information and have a probe that you can take to the field that tells you whether these genes are present and how abundant they are in contaminated sites, because with that you can make predictions. And now we have ideas how these different reductive dehalogenases are involved in different steps. So the question is, how do you develop a tool that is cheap, comprehensive, and sensitive? And we decided to use a nanoliter qPCR platform. This is a platform based on nanoliter qPCR developed by a company called Wafergen. These are chips that is this size, exactly the size of a Ghirardelli chocolate bar. And this contains more than 5,000 nanoliter wells. And you can deposit in each well a specific primer sequence that you can then use in a quantitative PCR assay. So either you can have one sample and 5,000 different PCR reactions, quantitative PCR reactions, run with that, or you have a few hundred samples and then run 10 different reactions on these. The quality of that, it's linear of 4 or 5 orders of magnitude. And that's huge. Moreover, we can follow now, because we know that these reductive dehalogenases are linked in the genome. We would expect that the abundance of an organism tracks with the a co-occurrence of specific reductive dehalogenases, because if an organism disappears, it's not only one reductive dehalogenase that disappears, but all the others that are linked to that. And since we can package up to 5,000 different reductive dehalogenases on this chip, we can essentially track populations of dehalogenating organisms. So with some bioinformatic tricks, we sort of grouped these reductive dehalogenases into groups that we can distinguish them with specific primers. And we use groups where we know there are reductive dehalogenases involved, where we know of a specific function, because otherwise we don't know what it means. So we use this system, and then we also use a different phylogenetic marker. For those of you who are working with phylogenetic marker, the Dehalococoides strains are very closely related, and based on 16S RNA gene sequence, you barely can distinguish them. But since we know the hydrogenase, specifically the HAP-L hydrogenase, is a key enzyme for the electron uptake and also conserved in the organism, we use essentially the diversity of the HAP-L hydrogenase as a phylogenetic marker to identify different Dehalococoides substrains, because the resolution that you get is much bigger. And then we also put this on the these primers on the chips. So with that, we can then study population dynamics. And this is a setup that we developed in the lab. It's a chemostat setting, so where you grow anaerobic chemostat, retention time of 40 days. So the organisms double once in 40 days. So we can study really physiology of slow-growing organism. And we have a chemical profile of how this reactor worked with lactate. We had TCE being fed. And then initially we had a full conversion to ethene. And then we had a vinyl chloride formed in the intermediate. But we really had no idea, was this one organism or two Dehalococoides, or what was the population biology. So we used our chip and the co-occurrence of different dehalogenases and the covariants to identify populations of Dehalococoides, which we never could isolate microbiologically, to see actually how it behaves in terms of the dynamics. So here's the chemical profile, and here are the nanoliter qPCR data. So again, here we have 4 orders of magnitude difference in sensitivity. So what we learned is that this culture is dominated, again, these are quantitative data, by a TCA-containing type of Dehalococoides. And we also know what other genes are involved in that. In fact, you can see those here because these dehalogenases just co-vary. And then we have a VCR-type Dehalococoides, which contains the vinyl chloride reductase. And you can see here that this organism goes from more than 10 times to the 4 to 10 times to the 2, sort of basically by 2 orders of magnitude. In this course, where we rarely see any variation here in the chemistry, but as you can see here, with the co-occurrence of other covariants of other reductive dehalogenases, we can really track this organism. We have a PCA-containing organism which is less abundant than these two Dehalococoides. And we also saw here, which was really exciting, for a short period of time, the occurrence and disappear, the rise and death of a BVCA-type Dehalococoides, which is also a vinyl chloride reductase, but it's a less frequently found. So we would have never seen this kind of population dynamics if we couldn't resolve the genes in the system. So we are now using this tool with an Italian oil company, Eni, to look at their sites and as they're contaminated and to make predictions and to follow the progress. Finally, in the last few minutes, I want to ask you, sort of coming back to the beginning. So we have here this unusual group of organisms. They're kind of extraterrestrial, right? So they have a very specialized mode of life. They're growing strictly anaerobic. They're growing with chlorinated solvents. Where the hell do these organisms come from? So, and we knew that this capacity for reductive dehalogenation was the result of horizontal gene transfer, right? All these genes were sticking here. So what is the lifestyle of Dehalococoides in the absence of anthropogenic contaminants? So to make progress in that, we noticed that in a completely unrelated field, chloroflexi, Dehalococoides-like chloroflexi were found in deep sea sediments. So my colleagues that study the oceans and the sediments and the rocks in the deep subsurface in the oceans, they actually found in their metagenomes fairly significant numbers of chloroflexi. So here, up to 16% of the Peruvian margin, this is the site here, just off the shelf of Peru, 16% of the total population were chloroflexi that were related to our terrestrial Dehalococoides. And that's presumably a relatively pristine environment. In the Bay of Bengal, we had like 18% of these chloroflexi also being present. That seemed like a good source to go to because there shouldn't be any anthropogenic contamination. And the idea is, if we can get hold of these organisms and look at their genome, we would learn something about the origin. We would not know how to isolate this organism because what substrates are you trying out? You can try, like Paige has done, halogenated humics, which is a great idea to generate a large library, but we don't know whether we hit the right substrates. So we decided to use single-cell genomics with material from these different sites. And we particularly worked with the 1230 site from the Peruvian margin and did single-cell genomics. So in this approach, you are isolating single cells from deep-sea sediments like 5 meters below a sediment surface. You remove the cells from the sediment material and then sequence the genome of individual cells that you isolate. So what we found is the following. So if you look more closely, what does it mean what these chloroflexi are? Chloroflexi is a fairly broad, phylogenetically and physiologically broad group. So here we have green, these are photosynthetic organisms. These are aerobic chloroflexi. And our anaerobics here are sitting right here. Here's a Dehalococoides, Dehalogenemonas. These are our chloroflexi. The terrestrial chloroflexi. If we're looking at the phylogenetic signature of the single cell isolates, well, I shouldn't say isolates, but the sequences of single cells that we obtain, they are clustering here. So all these names here we actually were lucky, we got a population, we got 2 cells with the same 16S RNA gene sequence, so we think that these are populations, so we could bin these genome sequences. Then we have here from another side other Dehalococoides. So in other words, there is actually a group of Dehalococoides type chloroflexi where we have genome sequences that are very similar to the Dehalococoides that we study. So what do we learn from that? Well, actually, let me skip this one. What we learn is that most of them do not contain any reductive dehalogenase. That was interesting. We do find reductive dehalogenases, actually that was on this slide, but these reductive dehalogenases that we and others find are actually of the class 1 type, the non-energy conserving reductive dehalogenases. We have never found a energy conserving 1-chloride reductase associated with any of these cells. So in other words, the business plan of these terrestrial Dehalococoides before they became dechlorinators must have been a different one. To make a long story short, we think these organisms have a strictly anaerobic metabolism based on reconstruction of a metabolic pathway based on the genes that we find in these sometimes 80%, 90% complete genomes, is that we have a metabolism which is based on hydrogen and on the Wood-Ljungdahl pathway. So in a sense, these organisms look like homocytogens, for those of you in the field. What makes us really confident about this is that in all of them, we find an RNF complex. And RNF is a complex that is found in strictly anaerobic organism that is used for energy conservation of low redox potential electrons. So this is the organisms can actually conserve energy from oxidizing ferredoxin at minus 500 millivolts with NAD at minus 280 millivolts intercellularly. And this energy is conserved to pump sodium across the cytoplasmic membrane. We find these RNA complex in these cells. That's the only mechanism that we find for energy conservation. And we find specifically the C subunit, which is actually this subunit here, which maps most closely to those RNF complexes that we find in homocytogenic bacteria. So the working hypothesis is that the previous life of a Dehalococoides, which is important for the degradation and remediation of chloroethanes in the environment, was actually a homocytogen in its past life. And the reductive dehalogenases could plug into this organism because of the economy of low redox potential electrons, which then simply diverted these electrons going from reducing CO2 to acetate, reducing chloroethanes to ethanes. And I think I'll skip this sort of, this is essentially the summary slide saying that we have come a long way for our understanding of how an important class of anthropogenic chemicals are degraded. We have been learning a lot about the biology of the organism as well as the chemistry of these enzymes involved, and we developed a set of useful tools based on this molecular knowledge that guides ultimately engineering efforts. So with that, I want to finish and thank the individuals involved. Anutaman did the protein chemistry work in the lab. Kosh developed, was a grad student, he developed the nanoliter qPCR chip, which I think, when I was talking with Bill this morning about the tracing of antibiotic resistance genes, I think this might be a great tool for that. And he did develop the chemostat after a system that was in our collaborators' lab, Lou Semprini and Mohamed Azizian. Svenja Lohner did some work on the reductive dehalogenases. Anna started the single-cell genomics, and the work is continued by Meva. You see here this individual here, Sebastian Behrens. And I should say the whole work of tracking populations of Dehalococoides organisms was started when Sebastian was a postdoc in the lab, because we isolated organisms, we did biochemistry, but we really didn't have any handle on how do you track populations. And to also really appreciate that population biology is really important. It's not the big differences between organisms, but population biology. And Sebastian really started that work in the lab. So probably should be more in bold. Yeah. And then other collaborators here. Troy Stich and David Bripp, great collaborators for the EPR spectroscopy, and Lou Semprini, environmental engineer in Oregon State University, a long collaborator on this project. Funding was from ENI, the NSF, and CW. So with this, I thank you for your attention. I'd be happy to answer any questions.
Great talk, Alfred. Thank you.
Yeah, great talk. So I had kind of a specific question about the DCA.
Yeah.
Sure. So with the DCA, where you say it was going to ethene, so it's
Right.
a dihalo elimination.
Right.
But I was looking at the Km, for that is much higher.
Yes.
And so I'm just saying, if you have V over K, right, it's so much higher for vinyl chloride. So if that's formed, that's going to go immediately. So could it be going through vinyl chloride and then doing the more reductive mechanism?
Yes. So you have a very good point, because you immediately picked up this Km of 1.3 millimolar for DCA is lousy, which basically means that it is probably not a natural substrate for this enzyme. Yet, I think the interesting aspect for us was that it has the capacity to catalyze that. So if it goes through vinyl chloride is intermediate, you essentially form a double bond. That's mechanistically interesting how this occurs. Whether this is a single step of a dihalo elimination here to form vinyl chloride, or if there's a step where you have only elimination of one chloride, we don't know. Environmentally, I don't think this enzyme plays an important role, but it shows us that in principle it is possible that enzymes have actually single and dihalo elimination activity. That's what I wanted to say.
All right. Thank you. I enjoyed your presentation very much. You talked about you used the term extraterrestrial with
Right.
Dehalococoides, which I thought was really cool. You never really talked about the source of the dehalogenases themselves. They still, after watching all this, and I don't know the area very well, they still seem extraterrestrial. Could you just comment where you think the ultimate source of those are coming from?
Yeah, so now realizing that this talk is recorded, I shouldn't have said extraterrestrial, because I just probably was not the best choice. but it still makes the point that this is very unusual biochemistry. We don't know exactly where these enzymes come from. There have been reports of enzymes, or at least genes, which have similar signatures, like 2 iron sulfur clusters and chlorinoids. And they're involved in, forgot exactly which synthesis, in epoxide actually removal. So it is possible that this chemistry, to have ultimately a cobalt-1, if it really goes via cobalt-1 as a starting point in the catalytic cycle. Cobalt-1 as a very strong nucleophile just happened to be a good catalyst for dehalogenation. In fact, Larry was one of the first who showed if you just take B12, no enzyme, just B12 and reductant, you can get reductive dehalogenation. So that's the chemistry. So the idea might be that this enzyme, which contains a chlorinoid and iron sulfur cluster out of a different biochemical context, metabolic context, happened to be good at catalyzing this reaction, which the chemistry already does. And then natural selection basically took care of this and then put these onto a genomic islands, and then the genomic islands moved. I don't know, did I answer your question?
Well, I just asked you to comment, so yeah.
Yeah.
Actually, I make
Yeah.
a comment on that. So I hosted David Lays, and so he talked about so this epoxide enzyme is in a biosynthetic pathway, and it's structurally analogous to the reductive dehalogenases. So he's gotten structures for both enzymes. So I don't know that that's the direct precursor, but they're evolutionarily related.
So I have 2 questions. The first is on your genomic islands. You showed, at least in a couple of genomes, there are some CRISPR arrays.
Yes.
Is there any evidence for viruses mediating the horizontal gene transfer?
There has been debate in the field whether or not viruses are involved, and there's no conclusive experimental evidence. There may be, or maybe the finding that you have CRISPRs on the genomic islands, I think is interesting because it allows that the organisms acquire phage immunity without being infected by a phage. That's pretty cool, right? I think the role of phage in gene transfer is an open question. There's no good experimentation on that.
And then my second question was, so we heard a lot today about dechlorination, but is there any evidence that these organisms are capable of dehalogenating other halogenated compounds like fluorinated or brominated compounds?
Brominated compounds, yes. Fluorinated compounds haven't been tried systematically. We don't think so. Paige.
I guess my question is pretty similar to the previous one, but I'll just poke a little further. How much is known about what stimulates the horizontal gene transfer? Have you been able to understand the conditions that stimulate that process?
No. So I think this is...
Thank you.
No, that's a great question. And I think from an environmental engineering point of view, that's what you really want to know. In other words, do I need to augment my site with organisms? Or if I know that the genes are there, just wait and mix, and natural selection will take care of that, right? That would be the implication of that. So therefore, it's a really important question to think about what is the rate and frequency. We even don't know what the parent is for this horizontal gene transfer. We don't know if it comes from another Dehalococoides, it's trivial. We want to know how it gets into Dehalococoides in the first place. And we don't know what the parent is. So I think it's a great question, but we haven't systematically looked into that.
I'm probably the most ignorant person in this topic in the room, so
Welcome
bear
to the
with
club.
me. So in the end game here is to ultimately be able to selectively farm as humans so that we can then inject them into sites, potentially?
I think that's up to debate. I mean, what this shows is that there's a lot of evolution occurring in different sites. And I should say these cultures from which we isolated the DNA, they have been adapted in the laboratory. So we actually don't know to what extent we evolved these populations that we study in the lab. You have to be open to that. But still there's apparent activity going on in situ. What the implication is, so on one hand, we have now organisms that you can inject into aquifers. If you know, because we know the genes and we know you assess your site in terms of organism abundance and gene abundance to predict whether you have the potential for complete dehalogenation. And then you can stimulate carefully by choosing carefully the electron donors. Or you can also, if you know that the genes are present but not in the right combination, that's where the nanoliter qPCR then comes into play. You can say it might be cheaper to wait and see if the organisms evolve in situ, and then that the activity takes up, then improves slowly over time. Was that your question?
Okay, any more questions? There's one back there.
I would like to contend with Randall. I'm more ignorant than you are about it. But I understand that all these sites where potentially these bugs could be used, these are very fertile food places for the bugs. Typically, nature is such that if there is food, there are also people or animals or bugs that come to eat that food. So why doesn't that happen spontaneously? Why these bugs do not occur in these places?
That's a very good question. As Page showed, actually in very nice experiments, you do find low levels of these dehalococordes in natural environments. They're just not very abundant. Probably because there is not much substrate flowing through the environment that meets their very specific metabolism, because abundance of naturally occurring organohalogen is fairly low. So you do find these organisms, but at really low frequency. Now you have a contamination which brings in, as you said, good food, but the organisms are still present in low abundance, and the organisms need specific electron donor hydrogen to get to grow. So in other words, there is a potential that the population can grow, but it hasn't done that because there's a mixing issue, that the organisms are not in contact with the bulk of the contaminant, which is in the aqueous phase. So if the organisms are associated with soil particles, then we have a spatial issue. And there's also an issue about electron donor access, because typically these environments are energy limited. They're kind of pristine, means there is neither electron donor nor electron acceptor.
So food is there, but not everything that they need is there.
There might be food, but there's no fog. And I don't want to go any further.
Good. Thank you very much, Alfred, and thanks for the presentation.
Thank you.